So can we ask people to reassemble from outside, or if they're not outside, if we can at least close the doors so that we can begin the session? Yeah. So one of the things that we wanted to be able to do before summing up this meeting is to have each of the moderators for what we refer to as the session 2 activities yesterday, which I very briefly summarized at the end of the day, to have each of those moderators give an assessment of the content of their session. And so I would ask that John WAGNER, Brian STROM, who very graciously has already stepped up, and Darrell Abernethy come up to the table here, and we will ask each of them to give a 10-minute synopsis of their topic, and then we will have an open session to discuss this. And what we're hoping that we're able to come out with is some very specific concepts, ideas, deliverables from the various topics that were discussed. As some of you probably may notice, Rob CALIFF, who led the session on clinical trials, is not in the room. He's not able to be here today, so I will try to summarize his information, although the summary was just provided to me a few minutes ago. So while John is giving his synopsis, I will review mine.
That's my cue. Great. So yesterday we had a really great session on identifying and developing meaningful biomarkers. Well, at least I hope you all thought it was great. I did, and we learned that there's clearly a confluence of efforts right now. We heard from multiple sectors yesterday, but that's actually only a subset of the various efforts that are working on biomarkers right now. So the subject of legislation, obviously 21st century cures. The FDA has been obviously up to their eyeballs in biomarkers since the Critical Path initiative and before. There's a NIH biomarker taxonomy effort, which is slated to read out any day now if it hasn't already. The FNIH Biomarkers Consortium is working is really ramping up to work on evidentiary standards in biomarkers. Brookings and the FDA, CIRSE, CPATH, the biomarkers, the National Biomarkers Development Alliance, even the IOM has been in the act, as you all know, with a report on surrogate endpoints about 5 years ago. And then all kinds of folks are getting in the act here. Just the other day, the conservative think tank, the Manhattan Institute, took a full-page ad out in the New York Times, basically lobbying legislators to work on biomarkers. Like, isn't that like totally random? But we heard from Mark Sallet at NIST, Sashi Amor at the FDA, Gabriella Leverzari at Pharma, and John Michael Sauer at CPATH. So we did hear from a number of different sectors, real progress there along many different lines. Am I taking too long, Russ? And sorry to tease you. And so we're done, right? I mean, that was all good. Everything was good. No. There's a lot of work that needs to be done, and I think that really did come out in the discussion. And I think that it was really interesting to hear on the panel a lot of consensus on some of the challenges that face the biomarkers, the lack of predictability for stakeholders with regard to an evidentiary framework, the amount of time it takes to work through biomarkers to get them to a point of of regulatory acceptance or decisionability are really big issues. There were a number of technical issues that folks talked about, but those are probably more easily navigatable. They're really science issues that are probably easier to tackle in a lot of ways. And we ended up the discussion time with some remarks around what pre-competitive collaboration really is. And again, there's obviously still some disparity about what is in biomarkers pre-competitive collaborations and what's out. I think that it does still really depend on your perspective. My perspective, which by the way is not necessarily the perspective of Takeda Pharmaceuticals International Company, is that biomarkers are like electrical plugs, and we should standardize them to every single extent that we possibly can, because that's really the only way to enable their widespread use. And I think that's really in everyone's interest. I think we are at a moment, and we heard a little bit about this yesterday, we're at a moment where we really are able to make some progress. So there is this confluence of efforts. In some ways it's kind of pulling apart, but in other ways it's coming together. And what I would ask is that we all involved in these efforts in regulatory science are part of the forces pulling in as opposed to pulling apart. And I think one very auspicious effort is with the Biomarkers Consortium working on evidentiary standards. That's probably a key gap right now. And instead of creating a new effort, supporting that effort is probably one of the best things that we all could possibly do collectively. Back to you, Stephen.
Thanks very much. So what I will briefly do before I turn it over to Brian is have the unenviable task of trying to summarize the discussion on the clinical trial session. And I would start by saying I thought it was a very interesting and dynamic set of presentations that we have. Just to refresh your memory, first we had Martin Landry talking about big data for randomized controlled trials, opportunities and challenges. Susan Ward then spoke about approaches to overcoming heterogeneity in the rare disease space. Charles Jaffe gave a very interesting presentation about the role of open APIs, and as I've learned, it's fire and not fear. Enrique Avalos talked about data aggregation across diseases and between stakeholders from the perspective of the CPATH folks. And my takeaway message from those various discussions is that we're really in a remarkable time period in being able to think creatively about the way that we conduct and the way that we assess clinical trials, and that the role of the large amount of data, the sources of those data, and the tools that are available, the computational tools that are available to us to be able to conduct analyses, are really at a remarkable point. And that the interesting approaches to the design and analysis of clinical trials is in a spot that it's never been before. And of course, the ultimate goal would be to be able to enhance the efficiency of clinical trials, to decrease the cost of clinical trials. But again, I will reemphasize what I said before, is that these have to be done in a way that you don't end up in some way, shape, or form compromising the ability to get to the answer that we're all looking for. I do think that there was a great deal of discussion during the subsequent discussion period about the quality of the information, the ability to curate the information, and as was discussed at some length, whose data is it in actuality, and therefore who should control and who should decide how that type of information gets used. And it's a very interesting discussion. I was particularly intrigued by the extensive discussion around the ability to curate the information so that you have a good sense of what the problems and the opportunities are from that particular data. And I think that this is an area that needs to be explored in much greater detail. I think at one end, you heard the discussion from Martin that when the data are large and quite robust, that it doesn't need to be pristine to be able to be effectively used and to get to an answer that can be quite similar to what you may derive through more traditional approaches. And then from Susan Ward about the way to especially when you're dealing with much smaller populations, to be able to control for the fact that not every patient has the same exact clinical course, and when you're assessing data over an extended period of time, you have to take that into account, and there are very creative and interesting ways to be able to control for that. So again, it's a really remarkable period in clinical trial design, clinical trial, the conduct of clinical trials, and the use of the phenomenal amounts of data that we have available to us. There certainly is a lot more to do to be able to most effectively be able to use this type of information. So I think with that, I will now turn it over to the next presenter, which is Brian Strom.
Thank you. We heard 4 presentations, and again, like the others, I'll very briefly summarize them and then draw some synthetic conclusions, expressing my own biases in the process. The first was by Rich Platt, who talked about the Sentinel system, a congressionally mandated system now approaching or exceeding 170 million people. Congressional mandate was 100 million to be able to do large-scale claims databases, claim studies, supplemented by medical records when relevant and available. Second was by Eric Horvitz of Microsoft Research, who had fascinating data to show us about how they could use their search data, the Bing search data and Google and other search data, to try to generate hypotheses about drug effects. Third was John Holmes from Penn, who described using online discussion fora in a similar way in terms of what could be elicited from them about new adverse reactions. And the third was by John Brownstein at Harvard, who talked about Twitter feeds and other social media as sources of hypotheses as well. I wanted to summarize, sort of synthesize it, of course, in a number of ways. The 2 prior sessions before ours clearly led up wonderfully with a number of calls for it would be nice to do a rigorous post-marketing surveillance that would be predictable and give good answers and reliable answers, and we could get drugs on the market sooner in the process, and so on. And of course, those of us in the field have been doing that for 40 years. It's not new. We've learned by a lot of our mistakes in the process, and I'll come back to that. What we have available now is massive data sets, Sentinel being the extreme, to be able to do that. Medicare is another one. We do studies in Medicare, 50, 60 million people. Back in the 1980s, my statistician used to tease me if I did a study of 1,000 patients, I'd use non-parametric statistics because that was small sample size. Well, now 100,000 patients is small sample size in our field. But keep in mind, these are structured data. These have denominators. The only time electronic health records are used are when they can be linked to claims data, so you have denominator information, or going to the UK, which actually has a health system, unlike the US. And so those of us who have shifted to using electronic health record data use UK electronic health record data, not US electronic health record data, for exactly that reason. What we see in Sentinel is an enormous opportunity for hypothesis testing, and they've been doing that now for 5 years, beginning to address hypothesis strengthening, the quick and dirty answers to hypotheses that come from elsewhere. And with their renewal, they're now going to begin to get into questions of hypothesis generating, which raise all sorts of methodologic issues. As you can imagine, with the risk of false positives when you have 170 million individuals and you theoretically can look at every drug and every outcome linked together is infinite. And I think it's important to keep in mind the issue of quality versus quantity that Martin talked about in clinical trials, that when you're looking at epidemiologic data are different. Quantity versus quality in clinical trials is a very reasonable trade-off. When you're looking at epidemiologic data with large sample sizes, you can get very precise measurements of wrong answers. If you do it incorrectly methodologically, you don't have randomization to balance it out. And so the circumstances are different, and you need to think very hard about the interpretability and the utility. And we've learned a lot about that through the years. And as programs like Sentinel move on to hypothesis generating, we have to keep that in mind. We will have endless thousands and thousands of hypotheses. We heard yesterday also about other approaches of novel new data for hypothesis generating, particularly the intriguing opportunity to use social web data on the web, social network data, and otherwise to be able to generate hypotheses. But my caveat is keep in mind, we already have lots of hypotheses. The spontaneous reporting system gets over 1 million hypotheses per year in its data. Our shortage is not of hypotheses. Our shortage is of rigor in exploring the hypotheses. So as we play with these other data, and I certainly strongly encourage it, I think they're really neat ideas, don't repeat the mistakes that we as epidemiologists made 30, 40 years ago when people first came out with case control studies, and case control studies were done in a non-population-based way, getting wrong answers in the process. You need denominators. In order to make conclusions, you must have analyses with denominators so you can look at rates. And you know, it's important to, as we play with the new tools, to think of them correctly as as a hypothesis generating, not as anything that is causal. And so I'll leave you finally with the trade-off, which in many ways is what Steve has been saying too, is the trade-off between innovation and rigor. As we play with new approaches to innovate, don't lose sight of the need for rigor.
Thanks very much. The last session yesterday was one that dealt with innovation and modeling and integrating information, especially disparate information, and Darrell Abernethy was the moderator for that session, so I'll turn the microphone over to him.
Thanks very much, Steve. So with the presentations, I'll touch on what I think the presenters were trying to get at, and then make some thoughts about where the implications of that might be and what we might think about in this space going forward. First were discussions about innovative trial designs, use of master protocols and Bayesian randomization, and some really very elegant and thoughtful approaches to new ways to create good trial designs for both common diseases and uncommon diseases. Secondly, various modeling methods for decision making and trial design were presented, and this set of constructs that Sandy Aller-Heiligen put forward have been very helpful and effective in optimizing development programs in not only her situation, but in other companies and at the FDA. Thirdly, the use of consortia to better understand disease, and I guess by inference to better understand the development of therapeutics for those disease processes, and some of these consortia efforts were described. So I would say that all of these activities are dependent on data development, its interpretation, and utilization for knowledge creation and innovative applications. And that being said, in my view, there are some serious gaps that we're at a great juncture to think about addressing. You know, first, let's think about the workforce. That was discussed briefly this morning. I think that it's useful to go back to an earlier step in thinking about workforce and think about who does what. So in my view, people who go into biological and biomedical disciplines are qualitative by nature. People who go into informatics and computational disciplines are quantitative by nature. And those are really quite different views of how you think about science. And so the challenge, I think, is to think through how to create situations in which people with those fundamentally different ways of thinking about scientific problems can synergize and collaborate with each other in effective ways to leverage each other's skill sets. So I think the training programs that encourage that sort of integration are key. And I think that they are wanting and absent, at least in the United States, for the most part, and that to fill that gap is a very important and exciting challenge in my view. Secondly, of the WHO, a number of us in this room, since I know many of you and we're approximately the same generation, came through at a time in which the best and the brightest from anything went into the biological, biomedical sciences. That hasn't been the case for quite a while, quite bluntly. And a real challenge is how to think through how in these areas we can, again, begin to attract the best and the brightest. Now, I know that I'm not the first person that has made this observation, but it's really going to be key to move the science forward in the best and most effective way. And then secondly, with regard to this group, how to attract these sorts of people into regulatory science. Because, quite frankly, if one looks across academic biomedical, biological science, at least through my career, for the most part, the thought of a career in regulatory science is considered kind of a dead end. I think that's becoming less so. However, quite honestly, there is still that sort of a sense, I think, in many elite academic medical centers. It's been interesting to me to see the variety of regulatory sciences programs crop up at esteemed universities and creation of curricula by people who, quite frankly, have a quite limited background in regulatory science and perhaps a somewhat limited understanding of that bridge. Now, I don't mean that to be too critical, but simply to say I think here's a real opportunity for essentially a new scientific discipline or the synthesis of old scientific disciplines to move a very exciting and interesting area forward and make it attractive to the best and the brightest. With regard to manpower issues, that was mentioned briefly this morning, but it's really similar in biomedical and pharmaceutical sciences in general, and that is when we recruit at the FDA, at least in the area that I work in, really most of our very talented recruits are coming from Asia and they're coming from other parts of the world because that's where the best and the brightest are going into the sciences, not in the United States. There are a few people from the US, but for the most part, elsewhere. Now, areas that are underemphasized that I think are real opportunities for regulatory science to evolve are the development of mechanistic approaches to inform population science. I've heard the word mechanism used once, I think, in the last day and a half. And I guess I find that unfortunate, but in my view, if we have strong pharmacologic and biological mechanistic understanding, then that allows interpretation of population science findings, and population science findings can inform mechanistic hypotheses and further mechanistic understanding. And to me, that's very much part and parcel of what should be happening in the regulatory science arena to better understand adverse drug reactions. If you'd say, how have we gotten here so that a conference like this is essentially a population science conference, I think it's understandable because really what's called evidence-based medicine is really driving much of what goes on in clinical, the evolution of clinical treatment guidelines and the rest, and that's fine. However, it really has moved toward a purely population science sort of discipline. And so I think here's a tremendous opportunity to resynthesize mechanistic-based science with population science to better inform regulatory science. So that's.
Thanks very much. So before I turn the podium and the program over to the co-chairs, Alistair and Martin, I would just summarize by saying I think that this is a very interesting and exciting time for regulatory science, not only in the medical product space, which is what we've largely been focused on today, specifically in drug development, but across the array of programs and products that FDA regulates. There are equally exciting opportunities on the F side of FDA, the food side of FDA, as well as in the tobacco arena, that I think will be able to make major contributions to not only public health, but the way that we conduct our regulatory activities. And I think that where we can go in the area of regulatory science is only tempered by how much we are able to invest in terms of the resources to do much of the work that I think that you've heard about over the last day and a half, but also whether or not we can have the type of quality people engage and participate in activities that will help inform the science. I will just end by saying something that I said yesterday, which is that we view FDA as being a public health agency that makes regulatory decisions through the best science. But in order to be able to do that, you need to have the best science. And I think that the opportunities are limitless. Certainly the types of products that we see coming into the door are using phenomenally creative mechanisms to be able to develop them. We have to be able to have equally phenomenal individuals that can actually understand that science to make the proper decisions regarding the review of those products. But all in all, I think that the potentially the sky is the limit in terms of what we can accomplish if we're able to make continued forward progress in the area of regulatory science. So again, I will just thank the organizers for the meeting. The focus has been on bioinformatics. I think it's very appropriate that that was the focus, because that's where a lot of the action happens to be these days. But I'm going to now turn it over to Martin and Alistair to try to at least come up with certain very specific deliverables that will come out of this particular workshop that will help us move forward.
Did you just pass me the microphone?
You're down first. Yes.
I think that's called being put in the hot seat. So joining the panel are Eileen Cannon, President of Pharma, and Mark Rogers, Board Chairman of the Reagan-Udall Foundation. And while Mark is joining us, Eileen, would you care to just take a few moments to reflect on what you've heard so far and where you think we can be most helpful to the forum in future-looking agenda items?
Absolutely. Thank you for this opportunity. I'd also like to clarify it. I'm actually just of the Pharma Foundation, so I can only speak for the Pharma Foundation. So the Pharma Foundation is a 501c3, and we will be celebrating our 50th anniversary next month, beginning next month. And our history has been to support young scientists in disciplines important to the pharmaceutical research and to encourage them to pursue careers in pharmaceutical research. Our board consists of R&D leaders of our member companies, and they are really interested in having the foundation help enhance regulatory science education and training. So we've been working on programs or trying to develop programs that will do just that, to work with our partnerships and to be able to support the young scientists in a way so that they'll be able to have better training and more educational opportunities to become substantial scientists in the regulatory science area. recently, well, last year, as Scott mentioned, we had a workshop that I worked with University of Rochester and Georgetown University, and a number of people in this room were part of it, and I thank you for your participation. Following the workshop, there was the article that was written that gave the findings of the workshop, and after that, I went to work with what were the recommendations from that workshop. Came up with a number of concepts that perhaps the Pharma Foundation could work on based on our focus on education, and just had a board meeting earlier this month, and they would like us to develop 2 of those concepts. One of them is to support rotations within industry and academia, and the other one is to support a mechanism, a way that senior scientists from academia could help the FDA in need areas based on whatever the FDA requires at the time, as long as it's within our realm of pharmaceutical research. And that program is really very fitting because we have a number of relationships in the universities, within the universities, because of our 50 years of support. And the way that will rely upon the FDA communicating with the Pharma Foundation as far as what the needs are, and then we'll put out a request via our website and have scientists apply for it. Everything that we do is peer reviewed, so that I have to build that program, but right now that's one of the things we're working on. As far as the rotations go, I surveyed the people that went to the workshop and a few additional people, had substantial feedback, and I divided the survey into 3 areas: academic, industry, and FDA. And it's our belief that the Pharma Foundation doesn't belong in the FDA as far as supporting the rotations there because of our relationship with industry. However, if I was going to pester these people for information, I might as well find out what they thought an FDA rotation would look like as well. And we're hoping that I can share that information with other organizations like the Reagan-Udall Foundation, and perhaps they can develop that component. But right now, I'm working on developing a rotation program for industry and academia. So, thank you.
Thank you very much. Mark?
Thank you. My background is quite eclectic in that I've trained in 4 medical specialties. I have an MBA. I do a lot of things, and my view reflects a kind of eclectic, long-range view of what regulatory science really means as I see it. I tend to think backwards from 20 years forward and think that what kind of people will we be needing to train for what purposes? I think the real truth is that there's a lot of difficulty in communication between different segments of society and different professional segments of society. And as a result of that, I think that we have to be a little bit more broad-based in terms of contemplating where regulatory science could go in the future, because we have to think about who should be trained in regulatory science. As an example, one of my colleagues has been dean at Mayo Clinic, and they see this of interest out of engineering and want us to think about people who would get involved in engineering and how they would do that. I have other colleagues who would say, you know, people in law may end up having to be trained in some fashion. There are a large number of people now who have MDs and JDs. And how are we going to train the future people who will succeed Steve and all the people at the FDA? And is it related just to the FDA? Or should some people, if you'll pardon me, even at the Supreme Court be trained in the issues that have to do with biology and law? So with that as a concept, that we want to train people who will be able to integrate in society a vast array of complicated issues, I would ask that we think not so much about the word science, but perhaps have a more liberal arts approach to what the possibilities are. The skill sets that people need will include knowledge of how the government works, how to negotiate things with the government, how difficult it is for organizations to make decisions. It will include legal backgrounds on what the current state of the law is, not just the science of how you get this manufactured, but how you get it approved in some fashion. And I think about that a lot because in my own experience, I'll just share with you that one of the other things I did was I went to Wharton in my spare time, and I got an MBA. And an MBA, they sat me down for several weeks and taught me about corporate law to make sure that I wouldn't violate the law as I was building my business. And that turned out to be very useful for me. So I don't want to perseverate here, but I think you ought to reflect on a much more eclectic, broad-based, liberal arts approach to include what you've covered in regulatory science, but think 20 years down the line is what the implications of the people who need to be trained are and what their role in society will be. Thank you.
So I sort of have taken three broad, maybe two broad perspectives from the last 2 days. The first one came from Sasha Moore. Is she still here? Maybe not. She remember talked to us about biomarkers, and she said that the biomarker studies they'd done, 24 of them had been done, and 20 of the companies were quite happy to share the data, and 4 weren't. So what that tells you, I think, is that we need to better define the pre-competitive space, and we need to broaden that pre-competitive space. If companies can't amongst themselves even define what that includes, then we've got a major problem. Why should we broaden the pre-competitive space? The reason, I think, is that we bring larger numbers of people and brains into work on defining mechanisms. That's the second time, Darrell, you heard that word. But if we don't bring and broaden the pre-competitive space and attract more eyes, brains, whatever you want to call it, into that space to think more carefully about it, we're going to continue to move forward in this very narrow and frequently unsuccessful way that we've had in the past. So that's the first thing, bring in the power of the crowd, bring in the power of basic scientists across all the platforms that we have, industry, academia, translational scientists, and that should not be a temporarily related or temporally confined a definition. The pre-competitive space would extend throughout the drug development cycle, including once the drug was in humans and in later development. The second question that clearly came up and was raised by myself and others yesterday is the question of who owns the data. And it seems absolutely crystal clear to me that if a patient participates in a study, it's utterly unethical to say that that data should remain secret. And the reason for that data remaining secret is to force repetition of the study by someone else. That seems to me to be so clearly unethical that we need to rapidly recognize that that's not the way we can move forward. The final thing I would say is I'd like to decode, without asking Steve to agree with me, something Steve said a moment or two ago, and that is there's a big push just now to allow drugs to be approved on the basis of the effects on biomarkers. And there's legislation in play to try and do that, and I think that's a terrible idea. There are numerous examples out there where biomarkers have not told us whether a drug was effective or not. And I think it's critical that we don't just get carried along by the enthusiasm for biomarkers to allow these to be erroneously extended to the drug approval process, where we know from history that there are plenty of examples where not only would they not have worked, but in some cases, HDL was mentioned by Garrett earlier, not only would they not have worked, but would have come out very badly. So I think our enthusiasm for biomarkers has clearly misled legislators into thinking that that's a good approach for drug approval. And I think we need to be clear that we don't think that's the case, if people agree. I won't ask Steve whether he really meant that, but.
Great example.
Right.
Yeah.
I very much agree with what Alistair just said, and I would also like to remind the audience of something that Jim Stevens said that echoes, I think, quite strongly what Mark just reminded us of, and that is that precision does not replace judgment, and that the science actually does not occur in a vacuum, but that there are many societal pressures, including big P politics and small p policies, and that science participates in this broader societal discourse. And so understanding why we do the science and the limits of that science in convincing people of one thing or another. So I think we can get caught in this internal loop of great precision, of a strong scientific understanding, and still drift away from the public, whom agencies like the FDA and the EPA are sworn to protect. With that, I think we can open up the floor to questions and comments.